Hello everyone. In this video, we are going to see about the scientists who have contributed to the field of microbiology. We'll look into some important scientists one by one, starting with Antony van Leeuwenhoek. Antony van Leeuwenhoek was a draper and a haberdasher by profession. That means he used to deal with items of clothes and sewing. But his hobby was lens preparation. So he was the first one to invent a microscope. He constructed more than 250 microscopes in his lifetime, with magnification of around 50 to 300 times more than the specimen. So his lens used to magnify the specimen more than 50 to 300 times. With his lenses, he observed and described many microorganisms and called them animalcules. So, whatever he observed through his lenses, he reported the findings to Royal Society of London in Dutch language, which were later translated to English and published in Proceedings of Royal Society. He was elected as Fellow of Royal Society by British Royal Society. The things he observed and discovered through his lenses were a number of seeds and embryos of plants and small invertebrate animals. He discovered the existence of spermatozoa and red blood cells. He observed muscle fiber striations and nuclei of fish blood cells and myelin sheath of nerve fibers. He provided confirming evidence for William Harvey's theory of blood circulation. He is considered as the father of bacteriology, hematology, histology, protozoology. So, the next scientist we are going to look into is Robert Koch. Robert Koch is considered as the father of modern bacteriology. He developed many isolation techniques for microorganisms. First, he tried to culture bacteria using cut boiled potatoes, which was not proved to be ideal. Then he tried gelatin, even that was not found to be ideal. Then he used agar as a solidifying agent, which worked for culturing bacteria. After which he developed many different media using agar, and then he developed many methods for isolation and study of different pathogenic microorganisms, especially bacteria. He isolated the spore forming bacteria Bacillus anthracis and showed that it causes anthrax. He isolated tuberculosis causing bacteria called Mycobacterium tuberculosis. He, he is the first person to demonstrate that the role of the bacterium as a causative agent of disease. He formulated certain criteria for proving that a specific organism causes a specific disease. These criteria are known as Koch's postulates. The first criteria is that that particular microorganism must be present in every case of the disease, but it should be absent from healthy organisms. That particular microorganism must be isolated and grown in pure culture. And that isolated microorganism, when inoculated into a healthy host, it should result into the same disease. And the same microorganism must be isolated again from that diseased host. So these are known as Koch's postulates. Now we are going to see about Edward Jenner. Edward Jenner was the first person to attempt on artificial immunization. He attempted this procedure against smallpox. So during his studies, he observed that milkmaids who often contacted cows having cowpox become resistant to smallpox. So what he did was he inoculated a healthy 8-year-old boy, James Phipps, with cowpox virus. This inoculation of cowpox virus caused mild symptoms in the boy. Then he inoculated the boy with smallpox virus. This inoculation did not show any symptoms in the boy. So he concluded that this process of inoculation of cowpox virus led to immunity against smallpox. Later, Louis Pasteur coined the term vaccine in honor of Jenner. Vaccine is nothing but any preparation of a weakened pathogen that is used to immunize against infectious disease. The next scientist we are going to look into is Dimitri Iwanowski, who is a famous Russian botanist. He worked on tobacco mosaic disease and tried to find out what causes this disease. He took the sap from the leaves of infected plants and tried to cause infection in fresh plants, in which he was successful. So he found that the sap obtained from the leaves of infected plants is capable of causing fresh infection in healthy plants. Next, he tried to find that the infectivity of the sap is because of bacteria or not. So what he did is he passed the sap through some sophisticated filters. These sophisticated filters usually do not let bacteria to pass through them. But he found that the sap retained its infectivity even after passed through sophisticated filters, with which he concluded that the infecting agents are not bacteria. Next, he heated the sap before applying the sap on the healthy plants. So he found out that the sap loses its infectivity when it is heated before application on the healthy plants. He called these agents causing the mosaic disease as filtrable agents because they are able to pass through the filters. And these agents are submicroscopic with a nature different from bacteria. The last scientist we are going to look into in this video is Louis Pasteur. Louis Pasteur is a French chemist and physicist. He is considered as the father of modern microbiology. He was one of the scientists who dismissed spontaneous generation theory and confirmed biogenesis theory through his swan neck experiments. He studied extensively on fermentation and he found some facts such as for conversion of sugar into alcohol, living yeast cells are required. The souring and spoilage of beer and wine was due to the presence of some rod-shaped organisms in them, which convert sugar to lactic acid instead of alcohol. And for fermentation, each type of microorganism requires specific environmental conditions. He developed a method of sterilization called pasteurization. He discovered aerobic and anaerobic microorganisms. He worked on quite a few diseases like pebrine disease of silkworm, anthrax, chicken cholera. He developed vaccine for chicken cholera and rabies disease. So that's it for this video. I'll see you in my next video.